Thank you very much for the opportunity. I'm a complete outsider in the community, but I think I'm not the only one. So I'm really happy to share with you some of the things we are doing. Since time is short, I will go first to the acknowledgements. This is work that we have done almost entirely in the biomolecular simulations group at the Institut Pasteur in Montevideo, in collaboration with Gustavo Seabra and Leandro Martinez, two guys from Brazil. The work I'm going to show now has been performed essentially by Martin Soñora, Matias Machado, and Ezequiel Barrera in my group. Let me just set the laser pointer. These are the organizations that paid for the research, and everything, as I said, was done at the Institut Pasteur in Montevideo, and Uruguay, which is a very famous country. Even Homer knows where it is. So we are this small country here between Argentina and Brazil, and so let's just going to the thing. I will just show some very basic features of about Zika virus. I will take profit, by the way, from the very nice presentation before. So we have more or less an insight already about flaviviruses. Very shortly, Zika is an enveloped positive single-stranded RNA viruses. There are no vaccines and no antivirals for that. Most of the people is actually asymptomatic, and the transmission goes through mosquitoes or vertically mother to fetus. And the thing that actually raised a lot of concern internationally when the Zika virus started, the epidemic started in South America, was that it was discovered that it can be sexually transmitted, and the virus can be found after even 6 months in the semen of infected male people. So actually, since it could be transmitted because people just don't realize they were sick. And killing the mosquito may not be just the complete solution to eradicate the virus. This, as it was already introduced, and many other flaviviruses have been resolved by cryo-EM. And despite the fact that I am a protein guy, this is the only protein figure I will show in this presentation. This is the heterodimer of the E protein, the stands for envelope, plus the M from membrane protein, that are found in 180 copies in the cover of the Zika virus. This is just a picture of the cryo-EM. Some of the features have been already described, but essentially what you can see here is that there is all this dense packed region which is occupied by the protein, and then the membrane here. And the goal of this presentation was not to go deeply into the dynamics of the protein, which is something we are also doing, but rather going into the dynamics and the structure of the membrane, which is rather difficult to observe. I mean, if you check closely in the electron density, what you will see is that you can identify the position of the heads of the phospholipids, but not much more than that. By the way, I haven't seen the structure that was presented previously, but I think there are some interesting correspondences here. Instead of using experimental methods, what we do to go and check for the dynamics of the virus and the membrane is a molecular dynamics approach, which is actually a multi-scale, which is based on a coarse grain approach developed in my group here and at the Institut Pasteur de Montevideo. I won't go into the details because there's going to be another presentation on Tuesday about this. But essentially, what happens in the simulations is that we go through an onion-like configuration in which we have different resolutions for different things. For all the things that are important to this presentation, we can have resolution at the single residue level, so single lipids and even beyond. So this kind of approach is, to the best of my knowledge, the cheapest way to get nearly atomistic resolution of a virus and its dynamics. As you can see here in this superposition, you can see something like green or whatever the color is, superimposed to violet. This is the density that you can derive from the simulations, and also the black shadow here corresponds to the density that you can derive from the simulation. So although I'm not showing you those results, the overlap between the density that you can get from the simulations and the experiment is really very good, and we can get over 90% of correlation. So you can really rely on the data. When we started to see these dynamics, actually we had the idea that the membrane was nothing but just a support for the proteins. However, we started to see some interesting things. And before going further, I just would like to mention that in our membrane, because since we don't know exactly the details, we assume that the composition of the membrane is POPC, POPE, and POPS, so phosphatidylcholine, phosphatidylethanolamine, and phosphatidylserine in a 631 percentage. This is an approximation to the content of the ER membrane, which is where the flavivirus get assembled. So if you start a simulation at this, which is the point 0, what you see is that there is a random distribution of the red dots, which corresponds to the heads of phosphatidylserine. The other two components are all scattered around. But if you run the simulation, what you start to see is that there is a clustering of phosphatidylserine near the transmembrane parts, and especially in the luminal side of the virus. And this just called our attention. What you see in blue here corresponds to this amino acid, which is not the amino acid per se completely conserved, but yes, the basic characteristics. So it is either a lysine or an arginine in all the flaviviruses. So in principle, we started to think that there's kind of something more than just a support for the proteins, but actually that there is a deep relevance in the interplay between the lipids and the protein. These amino acids, of course, as you can see, is highly conserved. There is an enrichment of phosphatidylserine in infected cells. So we went to go further into the protein-lipid interactions. To do that, what we did was undertake a comparative study. So we wanted to see what happens with the protein DNA interactions here in the Zika virus, but we wanted also to have a comparison with the flat membrane patch, another protein inserted in a flat membrane patch. For that we use phospholamban, which is a protein which is common in the ER membrane, and also we use a vesicle, an empty vesicle, which is more or less the same size of the Zika VLP. The first thing that we went to check was about the contacts between protein and lipids. And if you normalize that through to the number of lipids, what you see, if you compare the lipid protein contacts in a normal or a human protein with the proteins that are present in the envelope, is that there is a much higher number of contacts between lipids and amino acids in the membrane. So indeed, this tells you that the protein, the quaternary structure of a protein is kind of imposing some pressure perhaps on the lipids. So when you go to evaluate the areas per lipid, so the size of each that each lipid occupies in the membrane, and you do it following this comparison that I implied here on the top. If you go to see for phosphatidylcholine, which is present only in one leaflet of the membranes in these three examples, you see that there is an effect on the curvature, because phosphatidylcholine has a relatively large area per lipid. But then if you consider the other cases, the colors here corresponds to the colors in the naming that they used. So in gray you have the vesicle, in light blue you have the flat patch, and orange is for phospholamban, and purple is for Zika. So you see that there is a curvature effect that tells you that the phosphatidylcholine will have a large areas per lipid. Then when you go to the flat membrane, then there is a smaller size. Then when you include the protein, the size gets even smaller. And then when you go into the whole quaternary structure, things get much more crowded for lipids, and really you have a significant reduction of the areas per lipid. If you go then to see either POPE or POPS, the curvature effect is evident in the shift from this gray peak from here to here. So in the phospholipids that are inside the membrane, there is a significant effect on the curvature. And if you go again to see what happens with the lipids in the Zika membrane, then you always have the same effect in the compaction. So it is immediate to ask how much is the pressure that these lipids have to support to go to these extreme values of compaction. This is a kind of difficult question, and even considering that we are just doing computer simulations, the calculation is something which is highly non-trivial. As an alternative strategy, what we did is to apply pressure to a flat membrane patch like this one and see how much pressure we have to apply to obtain the same areas per lipid. So this is just a flat patch with 1 atmosphere, and this is, and then we run progressively different simulations until we arrive to a pressure, a lateral pressure, which would provide the same areas per lipid in both approaches. So the number or the amount of extra pressure that you have to impose to the lipid to get to the compaction that you see in the Zika virus is in the order of 51 atmospheres. So we had to add 50 atmospheres to the normal condition to get to areas per lipids that are comparable to those of the Zika virus. This for the structure. Then if you go to see the dynamics, this is a picture. I don't think you will be able to see the arrows, but actually each point here corresponds to the movement of one lipid. The important thing here is that when you see red or red. 3
more minutes, Sergio.
Sorry?
You have 3 more minutes.
Yes, thank you. What you see here is that you have a more reddish colors in the inner leaflet than in the outer leaflets, indicated that the diffusion or the instantaneous displacement in the inner leaflet is much higher than in the outer. And the same you get if you go to the bigger scale when you consider the diffusion coefficients. You have a tendency for the inner leaflet lipids to go to move faster than in the outer ones. You can also analyze principal component analysis. You should not waste your time trying to pick up a difference here, because from the pictorial point of view, this is an animation of how the lipids move, that there are not so many differences. If you consider now, this is only phosphatidylserine because of shortness. But in the first principal components, there are not so many differences. However, if you go and analyze the two interesting things that you can quantify from principal component analysis, one is what you see in the diagonal, which is the trace of the covariance matrix, and this is a quantification of how much they move. You see that in the case of phospholamban, you have a relatively smaller motion. Lipids in the outer membrane, I mean, in the internal motion they move faster than they diffuse, and in the inner leaflet they move like smaller. I mean, the internal thermal agitation and in the lipids in the inner membrane are smaller than the outer. If you compare how much they are similar one against the other in an inner product of the eigenvalues, or actually the eigenvectors that define the motion, only the outer leaflet is similar to the lipids in the case of phospholamban, while they are not so similar in between the inner and outer. So really, there is a decoupling of the motion between the inner and outer lipids. And this is essentially determined not only by the protein interactions, but by the protein plus the lipid plus the quaternary structure during the life cycle of the virus, or the replication cycle actually. It goes through partially mature and bumpy structures and breathing motions. And our idea now is that actually all this pressure which is stored gives the virus the opportunity to breathe and also to go from environments in which the pH is lower or higher in such a way that it can actually expand and contract without exposing the values cargo, which is the genetic material. Once it gets to the mature form, it is like kind of spring-loaded, and then eventually also that tension can be a part of the triggering and infusion mechanism. So the idea is that kind of shift the paradigms of proteins doing anything or everything in the lipid in the virions, but also putting the lipids and the membrane itself kind of in an important role in that. With that, I would like to stop, and of course, if there is time, I will be happy to take questions or comments. Thank you very much again.
Thanks, Sergio, for your fantastic talk. We don't have any time for the question now.